Hi students, today we are going to study about Unit 18 Heredity. We are all familiar with the term heredity. Heredity is the transmission of characters from one generation to the next generation. In our previous lesson, we talked about the process of reproduction. There we study what is reproduction, what is the need for reproduction, how the process of reproduction takes place, like that we studied. In this lesson, we are going to study how the new organisms are very much similar to their parents. You must have heard that people say you are like your mother, or your nose is like your father, like that. Many features that we possess, such as our external appearance, or body language, or even our habits, most of our portion comes from our parents. And also some of our characteristics might have been inherited from our grandparents. Not only humans, all other organisms which exist on earth have characteristics similar to their parents. How these characters are inherited from parental generation to the offspring? The reason is genes. What is gene? A gene is a basic hereditary unit which contains information for a particular characteristics. So where is the gene present? That we had studied in our lower classes. In our body, we have millions or trillions of cells. There are different types of cells present in our body. Muscle cell, blood cell, bone cell, like that. The shape or size may differ, but the basic structure is similar to all the types of cells. So, in each cell is surrounded by cell membrane. Inside, cytoplasm is there and nucleus is there. Nucleus is the controlling center of the cell. It is the important part of the cell. Inside the nucleus, chromosomes are there. In our body, the chromosomes number will vary from species to species. In our body, in humans, 23 pairs of chromosomes are there. The chromosomes consists of DNA. A small segment of DNA is called as gene. So, in our body, millions and trillions of genes are there. Each character is determined by a particular gene. So, we have millions of or multiple characters are there. So, multiple genes are responsible for determining their character. The branch of biology which deals with the study of genes, genetic variation and heredity of living organisms is called genetics. This is an important two-mark question. Genetics means the branch of biology which deals with the study of genes, genetic variation and heredity of living organisms. Heredity is the transmission of characters from one generation to the next generation. What is variation? Despite of being so much alike, any offspring does not like exactly similar to their parents. Even in your family, you may not exactly similar to your brother or sisters. There is a certain differences are shown between the individuals of the same species and also by the offsprings of the same parent. So, the variation refers to the differences shown by the individuals of the same species and also by the offsprings of the same parents is called variation. So, this heredity as well as variation can happen only due to these chromosomes. Gregor Johann Mendel is known as the father of genetics. He conducted a series of experiments on pea plants explaining heredity. He is an Austrian monk. He discovered the basic principles of heredity through his experiment. His experiments laid the foundation for modern genetics. He was born in a poor family of farmers in Silesia, Czechoslovakia. After finishing his high school, he entered into the monastery, Augustinian monastery at Brunn as a priest. From that monastery, he went to the University of Vienna for training in physics, mathematics and natural science. After return from the monastery in 1854, he continued to work as a priest as well as teacher in high school. During his leisure times, he started his famous experiments on the garden pea plant and conducted a lot of experiments for about 9 years, from 1856 to 1865. He had worked on nearly 10,000 pea plants of 34 different varieties. Mendel noted that many different varieties are there in this pea plant. Around 34 different varieties he found. Out of 34 different varieties, he had chosen only 7 pairs of opposing characters or contrasting characters of pea plant for his study. They are seed shape. He found some of the pea plants' seeds are round in shape, some are wrinkled. He considered the round shaped seeds are dominant over the wrinkled seed. And the seed color, some are yellow color, some are in green. In pod shape, some are smooth or inflated, some pods are constricted. In pod color also, some of the pods are green in color and some are yellow in color. Flower color, some pea flowers are purple color, some are white color. In flower position, some are in axial in position, some are in terminal position. Axial means flowers rises at the angle of the leaves. Terminal means the flowers forms at the terminal end, that is the end of the stem or the branches. Then plant size also, some of the plants were tall and some of the plants were dwarf. These 7 contrasting characters Mendel chosen for his experimental purpose. Then, what is the reasons for Mendel's success? Or why Mendel had chosen the pea plant for his genetic experiment? Because the pea plants are naturally very short lifespan, as it is an annual plant. Its lifespan within 1 year, complete its lifespan. So that is, he can get the result very fast. Then these pea plants are naturally self-pollinating. So it is very easy to raise pure breeding individuals. It is also easy to cross-pollinate. The flowers of the pea plants are bisexual, that means both the antecium and gynoecium is present in the same plant. As well as, apart from that, pea plants are found in varieties with contrasting characters. So because of these reasons, Mendel selected the pea plant. Before we are studying the experiment, Mendel's monohybrid cross, we should know about certain important terms used in genetics. The first term is gene. Gene is an inherited factor that determines a characteristic. So, each character is determined by a particular gene. In our body, multiple genes are present to control multiple characteristics. The next is allele. This allele as well as gene is complementary to each other. Allele is nothing but the different forms of the same gene. For example, if you consider the height is a character or trait. A trait is determined by a particular gene. The height has 2 different forms, that is tall and dwarf. This tall and dwarf is called as alleles. So, alleles are nothing but the different forms or the alternate forms of the same gene is called allele. Another example, if you consider eye color. Some people may have black color eyes, some people's blue color, some people's brown color eyes. So, eye color is a character. This character is determined by a particular gene. This black, blue, brown. These are the 3 different forms of that eye color. So, these black, blue, brown are called as alleles. If we consider the height of a plant, if the plant is tall, it means the tall allele expressed itself and not allowed dwarf alleles to express. So that The tall allele is called as dominant allele, and the dwarf allele is not able to express itself is called as recessive allele. So in process, if a plant is tall, its allele is capital T or capital T small t. If the plant is dwarf, its allele is small t. Why we are representing the gene allele as capital T and small t? Because we should understand one thing. The character height is a trait. It is represented by the letter T. That means it is the first letter of the dominant trait. That is the tall. The first letter is T. So the height, the character height should be determined by the letter T. If it is dominant trait, we should use capital T. If it is recessive, we should use small t. We know what is dominant and recessive. Dominant means the character expressed itself. So, if it is dominant trait, we should use capital T. If it is recessive trait, we should use small t. So, both the letter would be the same. We should not use for the recessive trait, since it is dwarf, we should not use the letter B. We should use capital T for the dominant, small t for the recessive. Like that in all other characters. So now let's study Mendel's monohybrid cross. Mono means single and hybrid means offsprings produced by the crossing of two different species. So monohybrid cross, this is the first experiment of Mendel considering the inheritance of single trait. So since it is the first experiment, he wanted to know how the traits are inherited in the subsequent generation. That is why he wanted to start with a simple thing. That is why he selected only one trait during his first experiment. If he selected multiple traits, the result would be confused and complicated. He would not receive the proper result. That is why in this first experiment, he considered single inheritance of single trait, that is the height of the plant. Mendel selected the tall and dwarf pea plants for his experiment. He observed their growth for nearly 2 years and found that tall plants always producing tall plants generation after generation, and this dwarf plant always producing dwarf offsprings generation after generation. Mendel named them as wild plants or pure breeding varieties. Now, he selected one pure breeding tall plant as well as one pure breeding dwarf plant and he crossed. In his F1 generation or the first filial generation, he got all the plants were tall. He calculated the percentage of tallness and dwarfness in that subsequent generation. So, in his first experiment, he crossed a tall plant, pure breeding tall plant and pure breeding dwarf plant. In the F1 generation, he got all the plants were tall. Now, he self-pollinate the F1 generated plants. So, these two plants, the F1 generated offsprings were self-pollinated, and in his second generation or F2 generation, he got the two different types of plants. Some plants were tall and few plants were dwarf in the ratio of 3:1. That means out of 4 plants, he got 3 plants were tall and 1 plant were dwarf. Now, based on these observations, Mendel concluded that certain factors are passed on from one generation to another generation. Today, these factors are referred to as genes. Mendel also found that the dwarfness trait not lost in the F1 generation but suppressed in the F1 generation and reappeared in the second generation. Mendel called these suppressed trait as a recessive trait and the expressed trait, that is a tall trait, as dominant traits. So, In his first experiment, Mendel selected a pure breeding tall plant and a pure breeding dwarf plant. The F1 generation he got, all the plants were tall. Even though one of the parents were dwarf, he didn't get any dwarf plant or intermediate plant in his first generation. In his first generation, he got only one parental trait, that is the tall parent. While he self-pollinated the F1 trait, in his F2 generation, he got the two different types of plants. That is, three plants of tall and one plant were dwarf. Now, in the F2 generation, he got the dwarf parental trait. So, he thought that this dwarf trait is not lost in the F1 generation. It is masked or suppressed in the F1 generation, but in the F2 generation, it is reappeared. That is why he concluded that certain factors are transmitted from generation to generation. Now let's explain the same experiment using Punnett square. Punnett square is a checkerboard that is a form devised by a British geneticist R.C. Punnett for the study of genetics. It is a graphical representation to calculate the probability of all possible genotypes of offsprings in genetic cross. In this Punnett square, we should have parental trait or paternal trait, that is a father's trait at the top and maternal traits at the left hand side. Now we shall fuse the gametes. So this is the parental trait. These are the gametes obtained from the, this is the gamete obtained from the father, this is the gamete obtained from the mother. So now let's fuse the gametes. When we are fusing the capital T with a small t, we will obtain capital T, small t. Like that, when this capital T is fusing with small t, we will get capital T, small t. Like that, here also capital T, small t, and here also capital T, small t. So in the F1 generation, all the plants were tall. Its genotypic genotype is capital T. Now, he self-pollinated the F1 offsprings, that is capital T, small t. So you can place the gametes. There is a paternal gamete, we can place at the top capital T, small t. This is the maternal gamete placed at the left hand side, capital T, small t. Now we can fuse it. When the capital T is fused with capital T, we will obtain capital T, capital T. Like that, capital T with small t, capital T, small t. Here, small t with capital T, again capital T, small t. When the small t is fused with small t, small t, small t. So in the second filial generation, he got the 3 plants were tall and 1 plant is dwarf. This is considered as phenotypic ratio. The phenotypic ratio, the phenotype means the expression of morphological appearance. That means the expression of observable characters. When we see the 3 plants were tall and 1 plant was dwarf, then genotypic ratio is 1:2:1. That means capital T, that is pure tall, is 1. Capital T, small t is 2. And this is also capital T, small t also, this is tall. As I said you already, it possesses one dominant trait. If there is one dominant trait, it is considered as tall plant. So, capital T, small t is 2. Then small t, that is the dwarf. There is no dominant trait is here. So, it is dwarf plant. So, the genotypic ratio is 1:2:1. Genotypic ratio is nothing but the genetic expression of a particular trait. So, the capital T 1, capital T, small t is 2, small t is 1. That is 1:2:1 ratio. Next, Mendel's interpretation of monohybrid cross. From this monohybrid cross, Mendel came to a conclusion and he said, certain factors are passed on one generation to another, now referred to as genes. These factors occurs in pairs, just like capital T for the pure breeding tall, small t pure breeding dwarf, and then capital T, small t heterozygous tall. Like that, these factors occurs in pairs. During gamete formation, these factors segregate. That means capital T is segregating as capital T and capital T. So during gamete formation, the factors segregate so that each gamete gets one of the two alternate factors. For example, the capital T, small t. This is the heterozygous star plant. During gamete formation, the capital T and small t, these two are segregate and enter into two different gametes. When F1 hybrids are self-crossed, the two factors separate and then unite independently, forming tall and dwarf plants. From this, so Mendel totally used 1064 plants. Out of 1064 plants, he got 787 plants tall and 277 plants were dwarf. His phenotypic ratio means the morphological expression of a particular character. His phenotypic ratio 3:1, that means 3 were tall, 1 dwarf. Genotypic ratio, that is his genetic makeup, genetic expression is 1:2:1. That means, as we said already, capital T is 1, capital T small t is 2, small t is 1. From this monohybrid experiment, Mendel proposed the laws of heredity, law of dominance. According to this law, when two homozygous individuals with one or more sets of contrasting characters are crossed, the characters that appear in the F1 hybrids are dominant and those do not appear in the F1 are recessive. So, as we said already, the character appeared expressed in the F1 generation is considered as dominant trait. In the monohybrid experiment, the tall character is the dominant trait. That yes, all the offsprings were tall. So in the F1 generation, dwarf characters were suppressed. The dwarf characters did not express. So that character which is expressed in the F1 hybrids are dominant and those do not appear or those do not express in the F1 are recessive. The next law is law of segregation. According to this law, he said the characteristics or traits of an organism is determined by the internal factors, now called as genes, which occurs in pairs. Only one factor from a pair of such factors can be present in single gamete. So these two laws he proposed based on his monohybrid experiment. Now, home fun. Students are asked to study the following questions and write as home test. First question, why did Mendel select pea plant for his experiment? Second question, what do you understand by the term genotype and phenotype? Third question, a pure tall plant, capital T is crossed with a pure dwarf plant, small t. What would be the F1 and F2 generations? Explain. Fourth question, flowers of the garden pea are bisexual and self-pollinated. Therefore, it is difficult to perform hybridization experiment by crossing a particular pistil with a specific pollen grains. How Mendel made it possible in these monohybrid and dihybrid crosses? Students, you can refer the guide and find the answers when you study. Thank you.